and welcome to our video lecture on topic D2.1 Cell and Nuclear Division. Our guiding questions for today: how can large numbers of genetically identical cells be produced, and how do eukaryotes produce genetically varied cells that can develop into gametes? Our objectives for today: we're going to review cell theory. We're going to talk about the roles and stages of mitosis, including the condensation and movement of chromosomes. We're going to describe cytokinesis. We're going to outline roles and stages of meiosis, also called meiosis. Both are right. Pick your favorite. We're going to describe meiosis as a source of genetic variation, define non-disjunction, and then wrap up with a summary of cell division by comparing mitosis and meiosis. Remember that cell theory tells us that all living things are composed of cells, and cells are the basic units of life. Cells arise from pre-existing cells. We're going to focus a lot on this today. Also, single-celled organisms carry out all the functions of life. There are 7 of them, and multicellular organisms have specialized cells. If you want to review, what about that very first cell that had to arise from a not pre-existing cell, you can go back to our video lecture on the origins of cells. We watched that last semester. So those cells that arise from pre-existing cells, they are generated via cell division. We've got quite a few different kinds of cell division. In prokaryotes, because they don't have a nucleus, we're going to call that cell division binary fission. So binary fission is cell division in prokaryotes. And us eukaryotes that do have a nucleus, we don't just divide our cells, we also have to divide the nuclei. And so we have a couple different kinds of nuclear division. We have mitosis and we have meiosis. In all of these different kinds of cell division, there is a mother cell, sometimes also called a parent cell. And then there are some daughter cells. If we're talking about binary fission or mitosis, there will be 2. In meiosis, we actually end up with 4 daughter cells. We're going to focus here on mitosis for a little bit, and then we'll come back to meiosis later in this lecture. The role of mitosis is continuity. Here we're looking at a micrograph of cells in an onion root tip. So we have lots and lots of rapidly dividing cells where we have lots of growth in that onion root. Each one of these cells is exactly the same as the parent cells. The number of chromosomes remains the same from one cell to the next. Because the role of mitosis is continuity, we're looking to grow or replace cells that are clones, genetically identical to their parent cells. That continuity is maintained by stages of a cell cycle. So we're going to talk about how we're going to go through these phases, but then we end up back at the beginning and we go through it all again, hence the cyclic part of the cell cycle. We're going to talk a lot about these phases in great detail on the next couple slides, but here's a quick overview. So we've got interphase. This is the normal functioning stage of the cell when the cell is just doing its job. And then these 4 phases are parts of mitosis. We've got prophase, metaphase, anaphase, and telophase. It's kind of like taking the PSAT, except it's the PMAT or PMAT. Again, in interphase, we have normal functioning. In prophase, notice that our DNA that was all spread out in the nucleus in interphase starts to get kind of clumpy. That nuclear membrane or nuclear envelope starts to break down also in prophase. Metaphase, you can remember metaphase in the middle. Those chromosomes get pulled by these spindle microtubules to the middle or the equatorial plane of the cell. In anaphase, we're going to pull apart these sister chromatids so that each new pole of this cell that is dividing has a complete set of all the chromosomes. And then in telophase, we're going to start to unravel those chromosomes, go back to more spread out DNA, reform that nuclear membrane or nuclear envelope, and then cytokinesis is going to come in and split the cell, the rest of the cell, not just the nucleus, into 2 chunks. And again, this is a cycle. So once a cell gets to that cytokinesis stage, splits into 2 new cells, those cells can each go through interphase. Interphase is crazy long chunk of time, and then through mitosis, and then those two daughter cells can again go through that cell cycle. Sometimes cells will exit the cell cycle. We call that cell cycle arrest or mitotic arrest. That can also be called the G0 stage, where it's not actively dividing. Sometimes our very specialized cells, the ones that are very, very differentiated, don't reproduce anymore. They just do their jobs and then die. A couple cool things that happen here in interphase. So again, this is just the normal functioning part of the cell. It's doing lots of stuff, making enzymes and proteins and growing. This is what happens in the G1 stage. G stands for gap, so this is the first gap, but definitely also think about G for growth, because the cell is definitely growing and functioning during these gap stages. So we're going to add more stuff to the inside of the cell. We're going to increase the cytoplasm. We're going to increase the endoplasmic reticulum and the Golgi apparatus, and all kinds of cool stuff is getting duplicated. During the S phase, S is for synthesis, we're going to replicate the DNAs. Remember all of those stages of DNA replication that we talked about, all those enzymes involved, the helicase and the primase and the polymerases. So this DNA replication happens here in the S phase. This is super important so that each of the new daughter cells has a complete set of DNA after division. G2 is another gap stage. So we have our second gap, but also think about G also helps us remember that the cell is growing. This actually is where the mitochondria and chloroplasts, if you're also a plant cell, are replicated. And what's super cool about mitochondria and chloroplasts, remember, because of endosymbiotic theory, we know that these guys actually replicate via a process super similar to binary fission. And so even though we're talking about eukaryotes, there is still just a little bit of binary fission involved. Once that cell gets through to the G2, the second gap stage, we're going to move into mitosis. Again, mitosis is prophase, metaphase, anaphase, and telophase. And then cytokinesis, we're going to divide the rest of the cell. Mitosis is all about the nucleus. Cytokinesis is the rest of the cell. Quick review of that DNA replication. Again, this happens in the S phase of interphase. Super cool thing about DNA replication, we know that we're going to start with a strand of DNA and we're going to replicate it, make a copy of it. Here we have that copied DNA, so we see that we have two strands. Super cool. These guys are genetically identical to each other. The same ATCG sequence here is also here. These two strands of DNA are held together by some super cool proteins called cohesion loops. These cohesion loops help to keep the DNA nice and organized. We don't want our duplicated DNA to shuffle away from itself, and then who even knows what copies of what chromosomes end up in each of the new cells that are formed during mitosis. And so this cohesion loading, these cohesion loops, just help to keep the chromosomes nice and organized, so that during the movement of all these chromosomes, we're moving things around the right way in a very organized way. Moving forward into mitosis, that first stage of mitosis is prophase. What happens in prophase? We're going to take that lovely nucleus. Oh, let me pick a different color so you can actually see it. We're going to take that lovely nucleus. We're going to start to break down the nuclear membrane, also called the nuclear envelope. So you can see this nice, lovely, clean-edged shape here does not exist over here anymore. That nuclear envelope, nuclear membrane is starting to break down. We're going to have these super cool centrosomes. Centrosomes are examples of mTOCs, microtubule organizing centers. Microtubule organizing centers. Centrosomes are one example of these mTOCs. Microtubules are these super cool spindle fibers, these super long, crazy protein fibers that bind to the chromosomes and then move them around a little bit. The other super cool thing that's happening during prophase, this DNA, again, it's all lovely spread out here, starts to get super clumpy. This is because the chromatin chromatin, which is that unraveled or uncondensed DNA, starts to wrap up into these super cool chromosomes. That process is called condensation. Condensation, the wrapping up, the bundling of the chromatin, that unraveled DNA strandy stuff, into chromosomes. Again, this is super important so that we can keep those chromosomes, that DNA, organized so that all the pieces go to the right parts of these dividing cells. So here's a close look at condensation, that wrapping up of the chromatin, our DNA strands, into some super well-packaged chromosomes. How this is going to happen, we're going to form some nucleosomes. Remember that nucleosomes are the strands of DNA and the histone proteins that the DNA wraps around. This is a simplified histone octet. These guys actually come in clusters of 8. That should be a 3-dimensional 8. Pretend like there's a cube of 8 histone proteins in there. So we've got that octet of histones, the DNA that wraps around it. Then those nucleosomes, the DNA and the histone proteins, the DNA is wrapped around, link to each other. So we don't just have nucleosome, nucleosome, but now we have these clusters of nucleosomes. And then these guys wrap around more and more. We don't actually really know how this happens. So if you are interested in molecular biology, this is an area of active research right now. Help us figure out, how do these chromosomes even wrap themselves up? Because we don't know yet, which is really cool. But eventually we do know that we end up with these lovely chromosomes, super organized packages that we can shuffle around during mitosis. Following prophase and that chromosome condensation, we're going to move into metaphase. Remember that metaphase means that we are moving those chromosomes to the middle. Those chromosomes are moved by these super cool spindle fibers. They're going to move to the equatorial plate of the cell. So again, this movement of chromosomes is done by these super cool protein fibers called microtubules. Microtubules combine to form the sum of the mitotic spindle. The spindle is just that collection of these protein fibers, these microtubules. The microtubules start lengthening from the centrosome. Centrosome is an example of an MTOC, microtubule organizing center. These centrosomes are going to get those microtubules organized, and then those microtubules are going to lengthen. They are going to bind to the chromosome's centromere with these other super cool proteins called kinetochores. The kinetochores with the centromeres and the microtubules will push and pull as those microtubules lengthen and shorten to get those chromosomes lined up in the middle of the cell during metaphase, metaphase in the middle. Those same microtubule fibers, the spindle fibers, are going to pull on the chromosomes during anaphase, and we'll talk about that in a couple more slides. This picture is super cool. Just a colored version showing the blue here is the collection of chromosomes in the middle of the cell during metaphase, and then all of these yellow and green guys are those different types of microtubules that make up the spindle fibers. So these microtubules, super cool. They can move those chromosomes around by lengthening and shortening, by growing and shrinking. They do this by adding and taking away these tiny little pieces. So this whole thing is called a microtubule. These little dimers, called a dimer because there are 2 pieces. Remember that di is our prefix for 2. So these dimers are called tubulin proteins. The tubulins can be either added to the microtubule fiber, or they can be taken away from the microtubule fiber in order to lengthen or shorten these fibers. The microtubule fiber, the spindle fiber, is going to attach with some super cool motor proteins. Here are those kinetochores that I mentioned before, the kinetochores that bind to the centromere and then also collaborate with these microtubules to push and pull these chromosomes around. These motor proteins are super cool, so they use lots and lots of ATP. That ATP helps them to change their shape, and that shape changing just moves things around. So being attached here, and the whole thing moves, and we can push and pull by changing the shapes of the motor proteins and lengthening and shortening the microtubules. We get these chromosomes to just move all over the place. It is kind of amazing. Those motor proteins and microtubules keep doing work in the next phase of mitosis, anaphase. So we talked about prophase, metaphase, anaphase, and then we're going to talk about telophase next. So here we are at anaphase. In anaphase, again, we have these microtubules. Here are our microtubule fibers. These protein fibers are now going to shorten. We're going to do that by removing some of those little pieces of tubulin. That makes the microtubules shorter because the kinetochores help to attach the microtubules to the centromeres of the chromosomes. Those chromosomes are going to get pulled toward these opposite poles of the cell as these microtubules are shortening. Here you can see they've gotten really, really short. Those microtubules are super, super short, and our chromosomes have moved away from each other quite a bit, again, to the poles of the cells. CS, that's just our central spindle. So this central chunk of microtubule fibers, the central spindle. The CF is for cleavage furrow. We're going to talk about that when we get to cytokinesis. Super cool thing happens in anaphase. We are going to break those cohesion loops that we added during DNA replication. Remember, quite a few slides ago, we talked about these cohesion protein loops that help to hold the replicated DNA together so that it stays nice and organized. During anaphase, we actually don't want those two pieces of DNA, those genetically identical replicated DNAs, we don't want them to stay stuck together. We want to send one copy to one side and one copy to the other. And so during anaphase, we are going to break those cohesion loops so that we can separate that genetically identical chunks of DNA, separate them to opposite poles of the cell. And here we finally arrive at telophase. Remember, we did prophase, metaphase, anaphase, PMAT. We have finally arrived at telophase. Here we are definitely showing our chromosomes quite far away from each other. The nuclear envelope, also known as the nuclear membrane, is going to start to reform. Our chromosomes are going to begin to condense. They're going to go back to that unraveled chromatin form. And here we've got our super cool cleavage furrow. So what happens is the cell membrane starts to pinch in. Got some super cool proteins that make this happen. And then the whole rest of the cell, the plasma membrane, so here we're talking about the nuclear membrane, but also the plasma membrane starts to pinch and pinch. This is our cleavage furrow. Here's a lovely image, diagram, drawing, whatever you want to call this, showing cytokinesis. Cyto means cell, kinesis is moving things. So here we're not just separating the nucleus. We don't just have nuclear division, but now we have the rest of the cell division. So cytokinesis is splitting the whole rest of the cell. All of our PMAT of mitosis, prophase, metaphase, anaphase, telophase of mitosis, was all about dividing nucleus, but now we're going to divide the rest of the cell. That's cytokinesis. Again, with our animal cells, we're going to have this super cool thing called the cleavage furrow. Cleavage furrow. This happens with some super cool contractile proteins that just squeeze, squeeze, squeeze in on the middle of that central spindle. In plants, it's kind of cool. So we have, again, our microtubules are doing lots of work. So these green guys are the microtubules. They're going to help to move some of these vesicles into place. These vesicles start to merge with each other to form what's called the cell plate. The cell plate will eventually merge all the way across the whole entire cell. And then we end up with after all of that merging, two different membranes. And then the cell is also going to start to send some vesicles that are full of pectins and other materials that lead to the formation of a new cell wall. So we have a cleavage furrow in animal cells. These contractile proteins are going to pull in on the plasma membrane and then eventually split it into two pieces. In our plant cells, we're going to have the formation of a cell plate due to the movement of lots of vesicles. Those vesicles are helped to the middle by those amazing microtubules, those spindle fibers that are still there. And then we end up with membranes and then a cell wall forming between those membranes. So amazing. Quick summary of everything that we've been talking about so far. So again, in interphase, we've got our nucleus. Let me pick a better color. We've got our nucleus, nice clear edges to that nuclear membrane. We can tell that it has not been broken down yet. The DNA is pretty well spread out throughout the nucleus. This is our G1, lots of growth and normal functioning. S, the DNA in the nucleus is going to be replicated. G2, a little bit more growth and functioning. This is also when we replicate our mitochondria in us animal and fungal cells, chloroplasts if you're a mitochondria, and chloroplasts if you're a plant cell. We move into mitosis. So here we move into mitosis. Mitosis starts with some prophase. We're going to break down that nuclear membrane. We're going to start to condense the chromatin that was in the nucleus into chromosomes. Lots of intermediary stages. We don't need to know that there's a pro-metaphase, but somewhere between prophase and metaphase, the nuclear envelope breaks down. Metaphase, we have moved our chromosomes to the middle of the cell. This is done with our MTOCs, our microtubule organizing centers that help to lengthen those spindle fibers, those microtubules, so that we can push those chromosomes to the equatorial plane or the middle of the cell. In Anaphase, we are going to shorten those microtubules, pull apart what we call the sister chromatids. I'm going to draw a chromosome super quick. So after replication, we have those two strands. Both of them are double helices. We've got these two strands of DNA, these two molecules of DNA. They've each got a centromere. This is a single chromosome when we're here in metaphase. But after the spindle fibers pull apart these sister chromatids, they're called sister chromatids because they are genetically identical to each other, that replicated DNA. Once they get pulled apart, once they're separated, each one is its own chromosome. So our sister chromatids are separated. Each is now its own chromosome. We're going to pull those to those poles. We have our central spindle here, the cleavage furrow where we're going to squeeze in with those contractile proteins. And then eventually we end up in telophase where new nuclear envelope or nuclear membrane starts to form. The chromosomes start to uncondense or decondense, and we move back into interphase and do the whole thing all over again because this is a cell cycle. What's kind of cool about cytokinesis, that splitting of the whole rest of the cell, not just the nucleus, is it can happen equally. We can have equal division of the cytoplasm. Here we have a paramecium dividing via mitosis, and both pieces are pretty much the same. Sometimes we have unequal division. Woo, if I could spell. Unequal division. This is yeast going through an asexual process known as budding. So here we have some yeast cells going through mitosis, and notice that there is not an equal division of cytoplasm. Instead, we have a fairly big cell and then a tiny little cell budding off of the bigger guy. It is still mitosis. I still have nuclei in each of those new cells. Those nuclei are genetically identical to each other, so this baby yeast is a clone of the parent yeast. They're just not the same size. Of course, this guy is going to eat lots of food and grow and end up big eventually. Another example of unequal cytoplasmic division during cytokinesis is during the super cool process of making egg cells or ova. This is not mitosis, though. This is now meiosis, which we're going to talk about next. Super cool thing happens here. We've got our primary germ cell, the stem cell, the oogonium, is going to differentiate into a primary oocyte. We have the first division of meiosis, and we're going to end up with one giant cell and one very small cell, this polar body. This is not going to form an egg cell. We're going to go through, are you ready for this, a second division of the nucleus and the cell, a second cytoplasmic division. And here again, we maintain most of the cytoplasm in one of the cells. The other guy is a polar body, is not the egg cell. And so huge mass in this one cell, very small masses in the other. And the reason is this: during fertilization, this egg cell combines with a sperm cell. The only thing that sperm really contributes is a nucleus, which means that all of the mitochondria and all of the ribosomes and all of the stored nutrients and ATP and endoplasmic reticulum, all of that needs to be in that egg. And so we load all kinds of amazing cytoplasmic material into one egg cell so that there's just a lot there to be ready to develop into an embryo, a fetus, and then whatever organism it's going to become. And again, now we're going to move into meiosis. Remember that the role of mitosis was continuity. In meiosis, we're actually looking to change things up quite a bit. In both of these guys, we're going to start with some diploid cells. Diploid means that it has a 2n number of chromosomes. There are pairs, not singles, but pairs of chromosomes. These pairs of chromosomes are called homologs or homologous chromosome pairs. These homologous chromosomes, pretty cool. You get one from each of your parents, so one from mom and one from dad. And they have the same genes on them, but not necessarily the same alleles. What does that mean? So maybe here we have one of the genes for hair color. Maybe from dad, you got the allele for brown hair color. From mom, you got an allele for blonde hair color. Both of these are genes for hair color. They're just not the same hair color. But they have the same genes in the same locations. That makes them homologous chromosomes. Same genes, same locations, not necessarily the same versions of those genes, though. Diploid, we're going to have pairs, one from mom and one from dad. At the end of mitosis, we have genetically identical, still diploid, paired chromosomes in the nucleus. In meiosis, however, we're going to change things up. We start with some diploid cells, pairs, but by the end of meiosis, now we have haploid. And so instead of having pairs, one from mom and one from dad, we have only one of each. So you either have a mom or a dad, or we're going to talk about this super cool thing called crossing over, where you get some chromosomes that are mashups of moms and dads. The role of mitosis is what we call equational division, because we have equal numbers of chromosomes at the end of the division. We need that to continue the growth and repair and replacement of cells. In meiosis, though, we have what we call reductional division, where we have reduced or halved the number of chromosomes in each cell. This is important because during fertilization, we're going to take one of those guys and then another kind of cell that was formed also during mitosis. We add those back together and we go back to diploid. Super cool thing. Notice that there is DNA synthesis. Replication happens in both mitosis and meiosis. So we copy the DNA and then we divide the cell, and then we divide the cell again, but we do not copy the DNA. There's no replication here, so that we do end up with actually halved numbers instead of the same numbers. What's pretty convenient about learning the stages of meiosis is that they are, for the most part, quite similar to mitosis. We've got PMAT. The only difference is we have PMAT 1s and then we have PMAT 2s. So prophase 1, metaphase 1, anaphase 1, telophase 1, these are also known as meiosis 1. And then we do it all over again. PMAT, prophase 2, metaphase 2, anaphase 2, telophase 2. This is meiosis 2, or the second division of meiosis. We're going to talk a lot about prophase 1, metaphase 1, anaphase 1 because they are super interesting, a little bit different from mitosis. In PMAT to meiosis 2, these guys are super similar to mitosis, so it'll be a quicker chunk of words, I promise. Prophase 1 is amazing, my friends. So this first stage of meiosis. We do not need to know all of these different names of the sublevels of prophase 1, but we do need to know that a lot of things happen during prophase 1. So just like in prophase of mitosis, we're going to break down that nuclear envelope. We're going to condense the chromosomes, but a special thing called synapsis. Synapsis is going to happen in prophase 1 of meiosis. This does not occur in mitosis. Synapsis is this formation of what we call bivalents. Bivalents are when the homologous chromosomes find each other. They find each other. They start to form some bindings, and then they trade pieces. And so you can see here how there's some winding, some overlapping of those non-sister chromatids, and then they just trade chunks. We call this crossing over. This is called crossing over. The reason is this. I've got this chromosome from mom and this chromosome from dad. Maybe both of these guys have again, let's say that we've got the gene for hair color. And from my mom, I've got maybe the blonde gene. And then from my dad, I've got that same hair color gene, but now it's the brown version of that gene. If we didn't cross over, then I might have a child that looked exactly like my mom, and that would be kind of boring. And so instead, we take chunks of my dad's chromosomes and we mash them onto my mom's chromosome. And then I take those pieces of my mom's chromosome and I switch for my dad's chromosome. And then we end up with these lovely recombinant pieces where I've got, this is maybe a chromosome that is all my dad, and this chromosome is mostly all my dad, but it's got a little bit of my mom mixed in there. So these super cool recombinations, this crossing over allows us to increase genetic variation amongst our offspring, which is super important for the survival of a species. Here's another diagram looking at prophase 1. Another super cool thing that happens is what we call independent assortment or random orientation of the chromosomes, the bivalents, during prophase 1 and then into metaphase. So here we've got our chromosomes, and maybe these are the ones, I'm being super stereotypical with the colors. Here's from my dad and here's from my mom. And maybe sometimes I might line up dad here, and then mom goes there and there. Whereas maybe another random assortment, independent assortment of those chromosomes, I could have 2 moms on this side and 2 dads on that side. So here we're showing just 4 total chromosomes. As human cells, we've got 46 chromosomes, 23 pairs of chromosomes, 23 chromosomes from our moms, 23 chromosomes from our dads, 23 pairs of chromosomes, 46 total. If I can arrange differently just with these 4 chromosomes, imagine the huge number of differences that we can get when we're looking at 23 pairs all lining up either over here or over there, lots of random orientation of these chromosomes. But wait, let's go back to that idea of crossing over. What if also we did some crossing over? So maybe I took like this chunk of dad and I put it onto this chunk of mom. And so now random orientation plus this crossing over, the number of variations of these chromosomes starts to get pretty wackadoodle, which leads to us humans being really pretty amazingly unique organisms, despite all of us having 99.9% of all the same DNA. So back An interesting mistake can happen during anaphase 1 or anaphase 2 called non-disjunction. In a non-disjunction, the chromosomes are not separating the way that they're supposed to. So in meiosis 1, anaphase 1, what ought to happen is that we, again, separate the homologous pairs. Each new cell gets one of those chromosomes from the pair. In non-disjunction, what could happen is that the pair doesn't separate, and instead one whole pair ends up going to one of the cells, and so then the other cell is lacking a whole entire chromosomes, and then this guy has a whole extra set of those chromosomes. During that second meiotic division, that second anaphase, we're going to split, and then these guys again have an extra copy, and these guys are missing a copy. This can also happen during meiosis 2. So maybe that first division, that first anaphase goes totally normally how it's supposed to, but perhaps in that second anaphase, anaphase 2, instead of separating those sister chromatids, those guys get stuck together and we end up with a cell that has one chromosome too many. This can result in a disorder known as Down syndrome. Down syndrome is also known as trisomy 21, because non-disjunction occurs with chromosome number 21. And as humans, we have decided to number our pairs of chromosomes, and so we have pairs 1 through 22. We call these our autosomes, our normal old body chromosomes, and then the last pair, pair number 23, are our sex chromosomes. And if we have two X's, we're most likely going to end up with some ovaries. If we had one X and one Y, then we are going to end up with some testicles. So trisomy 21, tri for 3, we have 1, 2, 3 copies of chromosome number 21 because of non-disjunction. We ended up with an extra copy of this chromosome. This non-disjunction can happen with other chromosomes, and we can have other disorders. Down syndrome is the most common of them because embryos, fetuses with this disorder are more likely to survive and be born. If we're talking about trisomy 2, notice how gigantic these chromosomes are compared to chromosome number 21. This extra amount of DNA would be quite fatal to the embryo. Most likely mom wouldn't even realize that she was pregnant if she had an embryo with trisomy 2, just because this would have such an enormous impact on development that the embryo would just not be viable. 21 is just a tiny little extra chunk of DNA. And so these offspring are definitely viable, but definitely have also some developmental differences. Let's just do a quick review of mitosis versus meiosis. Remember that we've got these homologous chromosomes. We've got S phase. We're going to have that DNA replication in both. We have PMAT only one time in mitosis. We have PMAT 1s and PMAT 2s in meiosis. We have equational division or equal numbers. We started off as diploid. We ended up with diploid. In us humans, that number is 46. In other organisms, other species, the number is not necessarily 46. In meiosis, we start with 46, that diploid number, but in us humans, again, other organisms have different numbers. In us humans, we end up with 23 chromosomes in each of these daughter cells, which is good because if I have an egg with 23 chromosomes and a sperm with 23 chromosomes and they combine in fertilization, we end up back at 46. And that would be better than having, say, 46 and 46. That turns into 92 chromosomes, and that's just too many. Let's take a quick moment to do an amazing Venn diagram. Mitosis versus meiosis. Another nice summary. So in mitosis, the role is continuity. We are going to replace cells, or we're going to grow with more cells. These are all going to be normal old body cells. Sometimes we call them somatic cells. That's different from meiosis, where the goal is some change. We want to have some genetic variation. In the cells that we produce, we call those cells gametes or eggs and sperm. We're going to start both of these guys with diploid cells. We're going to start, both of these guys are going to start with some diploid cells, but in our mitotic divisions, we go from diploid right back to diploid, and the daughter cells are genetically identical. We make some clones. We start with diploid also in meiosis. That's a commonality, but we go from diploid to haploid. So we have half the number of chromosomes in those daughter cells, and they are genetically unique, which is pretty fabulous for genetic diversity in our species. Both of them involve cell and nuclear division. So we have some cytokinesis in both, but here there's only one division, whereas in meiosis we've got two divisions. We're going to end up with 2 daughter cells. In meiosis, we could end up with 4 daughter cells unless we're talking about an egg cell, in which case you really only end up with 1 because all the other guys just kind of went away. Mitosis, there's no crossing over during that prophase. In meiosis, we do, we do have crossing over, which again is just where those homologous chromosome pairs, one from mom and one from dad, trade pieces of DNA so that we have, again, more genetically unique eggs and sperm cells. Both of them have PMAT, but again, only 1 PMAT in mitosis, 2 PMATs in meiosis. Both of them have DNA replication, or the synthesis phase of interphase. Both of them have interphase. Both of them have those amazing spindle fiber microtubule proteins that help to push and pull those chromosomes around. Good? So good. All right, before we call this done, we are going to play Name That Stage, just to practice a little bit. Name that stage of mitosis. Tell me, what do we think this cell, cell's stage is? Hopefully you're voting for interphase because it looks kind of just normal. How about this guy? What's going on here? Notice how we're starting to see some gaps in the nuclear membrane, and we're starting to see some clumps in the nuclear material. This guy is in prophase. Can we find a metaphase? Ooh, I like this one right here. Notice those chromosomes in the middle. Here's another metaphase in the middle. Here we've got some lovely anaphase happening, right? So notice how those chromosomes are getting pulled away from each other. And then last but not least, we're looking for some telophase. I like this guy right here. So notice how we still have a little bit of like bumpiness along the edges of those clumps of chromosomes due to that nuclear membrane not quite starting to form back yet, but we also have some cell plates starting to form, so we've got some cytokinesis happening in there as well. My guess is that this guy right here is a very newly formed pair of cells. That cell plate probably formed pretty recently here. Feeling good? Feeling good. All right, let's do the same thing now with meiosis. Let's name that stage of meiosis now. Look at these guys, both of them in the middle. So these must be some metaphases, but I feel like this one is metaphase 1 and this is metaphase 2. Why do I think that? Here I only have 1 cell, and so here we have those pairs of chromosomes lining up at the middle. Here we have lined up our chromosomes single file in each of the 2 cells that was formed at the end of meiosis 1. Let's see what else here. I think we've got maybe some prophase. Here's some prophase. Again, prophase 1, we've only got that 1 cell. Those chromosomes are condensing. Here I've got prophase 2. Notice that I've got 2 cells. Each of them, we've got some condensation of the chromatid chromosomes occurring. Let's see, let's pick. I want to do this one next. What's this guy? Definitely anaphase. Which one is anaphase 1? Which one is anaphase 2? Here we've got anaphase 1 and here we've got anaphase 2. How do we know? Literally 2 cells must be meiosis 2. Here we have our homologous pairs of chromosomes separating. Here we have the sister chromatids separating anaphase 1, anaphase 2. Last but not least, here we've got our telophase. Telophase 1, there's only one cell. Telophase 2, notice that I've got 1, 2, 3, 4 nuclei starting to form. Cytokinesis is going to occur and we'll end up with 4 gametes at the end of that last division. And my friends, on that note, we made it to the end of this crazy long lecture video. We reviewed cell theory. We talked especially about how cells arise from pre-existing cells. We talked about the roles and stages of mitosis. Roles of mitosis is continuity. Stages PMAT, prophase, metaphase, anaphase, telophase. We talked about condensing those chromosomes with the nucleosomes during prophase, the movement of the chromosomes by the microtubules from the microtubule organizing centers, the mTOCs, during metaphase and anaphase. We described cytokinesis, that splitting of the rest of the cell. Mitosis is all about the nucleus, cytokinesis is the rest of the cell. We have the cleavage furrow from those contractile proteins in animal cells. We have the cell plate formation from the fusion of all those vesicles in plant cells. The roles and stages of meiosis. Meiosis's role is change, which is different from mitosis, is continuity. We have PMAT1 and PMAT2. So super similar, but we just do it twice. Meiosis is an amazing source of genetic variation between the crossing over that occurs in prophase 1, as well as the independent assortment of the homologous pairs. During metaphase 1, we have lots and lots of different ways to put collections of genes into eggs and cells. Eggs and sperm. Define non-disjunction, we did that. Non-disjunction is when the chromosomes do not separate appropriately during anaphase, and we can end up with an extra or too few chromosomes. An example of a consequence of non-disjunction is trisomy 21, also known as Down syndrome. We summarized cell division with some amazing Venn diagrams. My friends, you did it. Great work today.